Hey everyone, today's guest is Dr. Jack Gilbert, who is the director of the Microbiome and Metagenomics Center at the University of California at San Diego. Today, Dr. Gilbert will present work from his lab on optimizing the microbiome of the built environment as an underrated aspect of health. So with that, welcome Jack, and let's get into the presentation.
Yeah, hi, Michael. Okay, so let me share my screen. Yeah, so I thought I'd give a brief presentation on how the work that we've been doing is enabling us to understand the microbial community in the environment that we most often live in. This is a social microbiome, as it were, because we are social creatures or creatures of sociality, and that social interaction has probably been driven by our need to share microorganisms, or I hope to convince you of that fact. So we live in a microbial world. We are constantly being bombarded by microorganisms. Our skin, our lungs, our gut, our any surface that's exposed or can be exposed to the outside world is being constantly engaged by microorganisms. They've been around for 4 point something billion years. We've only been around for 500 million years. So everything you can see with your eyes, plants, animals, etc., grew up or evolved in the context of a rich and diverse microbial world. And so the microbes have always been trying to colonize our bodies to either eat us or to form symbiotic relationships with us, where our bodies pump out things which they need, and they pump out things which we need. And the vast majority of microorganisms that are emitted from our body are quite beneficial. You will release around 30 million bacterial cells into your immediate vicinity every hour. And this is a basic understanding of what leads to a microbiome of a built environment. This system, which we've grown to adopt, if you will, this microbial milieu that's constantly around us, a microbial dust cloud, is controlled by our immune system. Our immune system is like a national park service or maybe a gardener. It's there to basically manage the microbial community, which lands on our skin, lands in our respiratory tract, we swallow into our intestines or our urogenital system. It's there to manage that community, to manage it, to make sure that the bugs that we want and we need and are beneficial to our health survive and thrive, and the bugs that aren't do not. So it's very much a selective system. We used to think of it more like an army. It's really not an army. It's much more like a gardener or a curator, making sure that the right bugs are there at all times. We now understand the microbial community to be one that's quite socially driven, right? So where do we get our microorganisms from? Well, we get them from the environment around us when we are born, and we are exposed consistently to the bugs from the environment as we grow. However, it's also from physical and social interactions between people. So a baby will get a lot of microbes from the mother, as well as some microbes from the father, and also from anybody the baby interacts with. And that microbial exchange, we believe, is vital to the ecological drive, the evolution of the microbiome inside and on that child. And then that constant interaction we have with people consistently shapes the microbial communities that live on and in our body. And we believe that many of the social behaviors which we and other animal species have evolved to have and demonstrate now are actually to promote microbial transmission. So think of things like grooming or co-feeding or mouth-to-mouth interactions, everything from kissing to cross-feeding, or maybe even nursing, the idea of suckling breast milk, and coprophagy in some mammalian species, such as mice and rats that eat our own feces. And even nowadays, we are actively exchanging microbes for clinical purposes through things like fecal microbiome transplant, or FMT, that allows us to shift the beneficial microorganisms from one healthy person into a sick person. We ran a study a few years ago now where we took 16,000 microbial samples, poop samples from nearly 600 baboons over a 14-year period. We had students watching to wait for the baboon to poop. They're running out and collecting that baboon poop and tagging who pooped it. And using this information, we're able to demonstrate that 97% of the bacteria that were present in any one of those animals were driven or associated with their social interaction. If you will, they were in part a hereditary experience, because the vast majority of sociality is driven by familial connections, i.e., your mother, your father, your sisters and brothers, etc. And so this interaction shapes that microbial transfer down the generations. But what we identified in these baboons was that shared bacteria were predominantly associated with things like fermenting dietary fiber or producing beneficial products that aid the health of the baboon. So the majority of the bacteria that were transferred due to the social interactions between these baboon populations were beneficial to the health. And their presence in abundance was predicted by that social group behavior, such as grooming between a dominant female and a recessive female. And we see the same thing in people. So in a study that we ran nearly 15 years ago now, we were able to identify microbial transfer between people living in a home. So here we have three people, a grown adult living with his parents in a home, and their feet, their noses, and their hands here in red and pink. We also have two dogs that live in this house, three dogs, in fact, and their feet and noses are found in this network diagram. And then we have surfaces like the bedroom floor and the kitchen floor, which essentially allow for microbial transfer between feet, and then things like light switches and counters, which have a more hand-based microbial transfer. And we can watch, as we looked at this environment over time, we can see the direct transmission of microbes in this space. And we can see that even we're sharing and interacting constantly with the microbes of dogs. In one study from several of my colleagues, they demonstrated that a couple that have a dog in their home share more microbial similarity between each other, i.e., they are more microbially similar than couples that do not have a dog, or couples even that have a baby. This suggests that the dog may be acting as a microbial transfer between people, allowing for more microbes to be shared with your partner. And we know that dogs make excellent transmission vectors then, and people in the same household typically share around 12% of their gut microbial community. What does this mean? Well, a lot of the time, being inside a building is allowing for physical interaction. So in this particular population, we had a young couple who had a lodger to save money on their apartment costs, living with them. And here we can see, when we look at all the microbial samples in this diagram, which is a principal coordinate plot, every single dot is a microbial community taken from one sample from an individual. And when we compare them, the young couple who are in blue and red are much more microbially similar to each other than they are to person 3, who is that lodger. And that is true for the hands and the feet. Often the samples over interact or are much more similar to each other in the hands and feet of the couple than they are of the lodger. Interestingly, the nasal cavity is quite unique, irrespective of who you are, probably because it's much harder to get physically interacting up your nostrils. So this dynamic means that when you live in a space, you are sharing microorganisms, of course, because you're breathing them in, they're interacting on your skin. But realistically, the biggest driver for microbial similarity is that physical interaction with another human being. And pathogens have learned to, if you will, take over these transmission pathways, right? Pathogens come in and they've taken over the beneficial transmission pathways of beneficial microorganisms, and they are the rare occurrences where an organism has learned how to take advantage of those transmission pathways to infect you. And this is everything from viruses and bacteria and fungi, which we breathe in and breathe out into that built environment, or things like bacteria, viruses, and fungi that we pick up on our skin and then we transmit back to the environment from our skin. And so these are really important concepts to understand, and everyone who went through the COVID pandemic will understand that that breathing interaction was vitally important to the transmission of the pathogen, whereas picking up from surfaces didn't appear to have much interaction. So figuring out even pathology and epidemiology of bacterial, fungal, and viral pathogens is important when we understand the communities of the built environment. We can also see when we go into an environment that colonization, that tracking of microbes over time is actually quite dependent upon who lives there. So we ran a big study where we looked at this brand new building, which was built on the University of Chicago campus in around 2012. And we were lucky enough to be able to get into this building as it was being built and before it became an operational hospital. You can see the lobby, the sky lobby here is on the fifth floor, and then we have patient rooms up here on the top and laboratories on the bottom. We focused on these top 4 floors, looking at patient rooms and that lobby environment to understand how that microbial community changed when a hospital became an operational hospital environment. And what we were able to show was that, yes, it became much more similar to humans, but that the interaction between people and within a person was entirely dependent upon how people worked in that environment. So here I have a staff member over here on the left. This is their hand, their cell phone, their shoe, their nasal cavity, even their shirt hem and pager. And we can see the transmission of things like Acinetobacter, Corynebacteria, Staphylococcus, Streptococcus between all these surfaces as the person touches themselves. We can also see that the staff is transmitting through their hands mostly to the armpit and the other patient's hands, bacteria such as Staphylococcus, Corynebacteria, and Acinetobacter as they physically interact with that patient. But what we don't see is any real transmission of bacteria from the patient back to the person, the staff member. And this, we hypothesize, is because that staff member is washing their hands after physically interacting with the patient. So they may be a transmitter to the patient, but they're not necessarily going to be picking those bacteria up and transmitting them elsewhere because they're washing their hands. This points to the value of hand washing, right? It doesn't matter if you go into the garden and you get your hands dirty, you're going to get a lot of immune stimulation from interacting with the bacteria and the fungi in the soil on your hands. But you're not necessarily going to see much transmission the other way. As long as you wash your hands after you've done that, you're going to get the benefit, and hand washing is also absolutely fine. We even went into a college dormitory, and just for fun, we were able to really identify that microbial transmission network and actually look at the social dynamics of a college dormitory for all of these students living in there. And we could tell who was physically interacting with whom and which rooms they were sharing, and for the most part. But this was mostly dominated by hand-to-hand transmission as people interacted in that space. We found this to be compelling, although we could not necessarily tell everybody everything that was going on in that dormitory for good reason. The problem is we've lived now in this kind of sterile, polluted world, right, where we live in these buildings. We have polluted air, we have social inequity, we chemically clean these environments. We have pollution and a low diversity of environment around us. It's mostly built environment. And that actually limits the microbial diversity that we're exposed to. That kind of microbial diversity you would be exposed to if you lived in a rural community, interacted with a lot of animals, interacted with a lot of plants, had clean air access. That dynamic is shaping our health. And this, as an illustrative example of this, we went into 2 relatively rural communities, but who had made a significant choice about how they interacted with that space. So the Amish community that we predominantly worked with in Pennsylvania, Pennsylvania, and this was Indiana, predominantly live on their family farm. So children grow up physically interacting with the farming environment. Their front door is like 20 feet, 30 feet from the barn door. And they work on the farm with the animals and with the farming environment before and after school. And the Amish have very, very low rates of asthma in their population. Whereas the Hutterites, who live in South and North Dakota, this population, they tend to have a housing community that's separated from the farm. So the children, especially all children under the age of 14, do not actually go on the farm. They don't interact with the farming environment at all. And the Hutterites, even though they have the same genetic predisposition to the development of asthma, they have a much higher rate of asthma in their population. So the Amish, who live on the farm, have around 4% asthma in their population. The average for the US is around 8%. But the Hutterites have almost a quarter, so 25% of their population can develop asthma in their lifetime. And we think this is due to the interaction. So to the interaction with the environment, to figure this out, we went into their immune system. And I'm not going to give you an immunology lesson here, but we have two real cell types here. We have neutrophils, which I like to think of as sentinel cells, the scouts running throughout your body looking for foreign objects. When they see a foreign object, they latch onto it, they send out a signal, and then white blood cells come along and they munch up everything, the neutrophil and the foreign object. If you're exposed to lots of foreign objects, like you live on a farm where you're breathing in lots of dust from the animals and the soil, you need a lot more neutrophils. And we see that in those Amish children. They tend to have a lot more neutrophils than the Hutterite children who are separated from that farming environment. The Hutterites have a lot more eosinophils. These eosinophils are pro-inflammatory response cells, and they can be associated with asthmatic interaction. So Hutterites tend to have a lot more eosinophils than the Amish. But the interesting thing for us was that these cell surface markers we found on the neutrophils, like CXCR4 and CD11B and C, were much higher in the Hutterites. What does this mean? Well, these cell surface markers are like proteins that stick out from the cells of the cell surface in the neutrophils, and they can actually make the neutrophils kind of sticky. And the neutrophils, if they have a lot of these cell surface markers, which generally happens when the neutrophils are older and haven't been used, they kind of stick together and they can form these rafts. And that raft might potentially lead to a significant inflammatory response when it finally sees a foreign object. So if your neutrophils aren't used very much, like the Hutterite neutrophils aren't, they get old, they get sticky, and they can cause inflammatory reactions. To prove this, we took mice and we made them sensitive to a common dietary protein of albumin. So when this animal gets of albumin, you can see it gets a significant increase in inflammation of those eosinophils this is a bronchoalveolar lavage. This is what's happening inside their lung. So their lung has a lot more of these pro-inflammatory eosinophils. But if we take that same animal that's sensitive to albumin and we expose it to dust from an Amish bedroom, from the Amish children's bedrooms, that the Amish children are breathing in deeply while they're sleeping, and then we expose it to albumin, we see that inflammatory reaction goes away. So no longer get an eosinophil burst inside their lungs. However, Hutterite dust doesn't have the same effect. So there's something about the dust that the children are breathing in that farm environment that is helping to significantly reduce this asthma-like inflammatory response. This is important, right? So children that grow up on a farm in the US have on average, about a 50% reduction in the likelihood of developing asthma. So we know that farming environment can help to reduce asthmatic interactions, and it appears to be bacterial and fungal antigens that are present in the soil and in the animals that are getting into the lungs and into the skin of these children, helping to reduce their allergic reaction. So can we bring the benefits of environments such as forests and farms to people? Well, I like to think of this in terms of trees. Trees are really valuable, not just because they can improve your mental health, reduce energy bills, improve air quality, support biodiversity, etc., but also because they themselves have a deep, rich microbial environment around them in the soil, in the tree, and that microbial environment can help to stimulate the immune systems of people that interact with trees. So putting trees into urban environments, for example, can not only help to beautify the ecosystem and provide shade and reduce stress, but they can also help to stimulate antigenic microbial interactions that can benefit people's health. And one great example of this was a group of scientists in Finland who went into children's preschool children play groups in the cities and brought soil from the forests outside the cities and allowed the children to physically interact with the soil in a randomized way. The kids that physically interacted with the soil from the forests had a significant reduction in their inflammation. They were much less inflamed. Their immune system was much healthier than children who did not physically interact with that soil, suggesting that that soil environment can be beneficial. Working with Professor Amber Fife Johnson from the University of Washington, we're examining this phenomenon in something called the COMETS cohort, the childhood obesity and the role of the gut microbiome, outdoor time, and stress in the development of their inflammatory milieu as these children grow up, by taking children from urban environments and allowing them to experience farm schools or forest schools, so that they can get that immune benefit early in life and hopefully reduce their cardiometabolic disease rate. So these are ongoing programs that are predicated on the fact that kids actually really like being in forests, being outdoors, being at farms, and they can learn a lot, not only in their brains, but also in their immune systems from being exposed to these diverse networks of ecosystems. So I like to propose that we need to figure out better ways of rewilding our lives. And we're doing this by developing microbial building materials where we actually take bacteria and we build them into the structures that we use to build our built environments. Creating also living walls that contain microbes and plants to allow for improved immune activation of people breathing and working in that space. Increased ventilation is also valuable if you have a beneficial microbiome outside your house. And even multi-generational living could be improving, right? Moving from this very suburban, wealthy environment where we live in our little bubbles to more interacting with each other on a much more day-to-day basis. For example, we see that lactobacillus, a bacteria which is generally associated with health in our lung and on our skin and in our gut, can be at greater proportions in people who are infants who have older siblings in their home, potentially because those older siblings are interacting with the wide world and bringing that wide world back into the home for the children. We're also adding bacteria, working with our colleague Richard Beckett at the University College London, into public art. So these are ceramic surfaces which are impregnated with living bacteria, and then we put them into to open spaces and actually encourage people to physically interact with it, with the idea that when they physically interact, their immune system gets stimulated on their hands and in their bodies and helps to improve their health. Thank you very much to these are all the people that sponsor our work. And this is my team in what's usually sunny San Diego. And if you're interested in gastrointestinal health, please watch our documentary, Hack Your Health, The Secrets of Your Gut on Netflix. Thank you.
Thanks, Jack. That was great. Questions? So I have a whole bunch, so we can hopefully get into some of the specifics, right? So in just starting with the social microbiome, the shared social microbiome, I guess you can make an argument for choose your friends wisely, right? Because if you live a healthy life and the majority of your friends are eating unprocessed foods and, overweight, all the things that are associated with a bad microbiome, that would be transferred to the healthy, right? And you could make the argument vice versa. The unhealthy want to be around the healthy, but the healthy wanting to keep their health associated microbiome, right? So there's a balance there, I guess. What are your thoughts on the choosing your friends wisely?
Yeah, I think the complexity here is like it's much easier to change your microbiome when you're an infant, when you're a baby, right? And so a baby doesn't get to choose their friends wisely, as it were, but their parents' microbiome and the microbiome of the children and the microbiome of the people that baby interacts with should be healthy, right? Then you'll get that beneficial transfer. There's a small anecdote here. So babies that tend to exist in developed societies like China, Europe, North America, they tend not to have a certain bacterial species called Bifidobacterium longum infantis in their guts. This bug has essentially evolved to just consume all the sugars in breast milk, right? But babies that get breast milk should be able to consume those sugars based on having this bacteria in their gut. But if you're missing bifidobacterium, you can't digest the breast milk properly, and you don't get all the benefits of that breast milk. Where did this bug go? Why is it not present in our societies? We definitely know it was there in the 1920s and 1930s because we have records of people culturing it out of stool. But its prevalence has dropped to virtually zero or very, very small numbers, maybe 10% of our population. We think that's due to multi-generational antibiotic use and poor diet, leading to mothers and their daughters daughters not having those bacteria present inside their guts and therefore not able to pass it on to the children. So it's a possibility that we need to intervene with that, bring that bug back into our environment, back into our children's lives in order so that they can get the benefit of breast milk use. Later in life, you want to be exposed to beneficial organisms. And so being exposed to people who are healthier may help. but also just getting outside and gardening and going on a forest walk and living, with a dog maybe, or living with that interactive, diverse microbial environment could be just as beneficial as an adult.
Nice. All right, wait. So the bifidobacteria story, is that in part related to less breastfeeding too? Are mothers breastfeeding less on average?
Mothers are breastfeeding less, but that's really, we don't think that's the primary driver. We think it's the fact it's missing. So a lot of women that do breastfeed that have this bacteria in their gut, it ends up in the breast duct. So it's actually breast milk can be what we call a symbiotic. It's part probiotic. It's got the living bacteria in there, and it's part prebiotic. It's got things which will feed those bacteria and help those bacteria to produce things which are beneficial for us. So breast milk is like this ideal, living bacterial, almost like a kombucha, right, or a fermented milk product, right? It's perfect for baby's intestine. But if that bug is missing from the mum's gut, it never ends up in the breast milk. So we think, you need it in the mother's intestine in order to be able to give it to the child. So it's less necessarily about the lack of breastfeeding and more about it's missing from her gut.
And so then that goes to the antibiotic story. So more people than ever probably using antibiotics, less women having it in their gut, then it's less in the milk. The babies get less, and then you have the multi-generational. So I should say I've had my gut microbiome done, I think, 3 or 4 times, and almost no bifidobacteria. So I'm in that, same boat, unfortunately.
Well, so when you breastfeed your children, you want to be careful. But yeah, I mean, like the fact it was prevalent in our society back in the 20s and 30s before the start of antibiotics would suggest that hypothesis, right? Antibiotics really only came in the late 40s, early 50s. So it's indicative of that association, but more research would need to be done to make that definitive statement. It's also quite possible that poor diet leads to a refined diet leads to a reduction in the abundance of some of these key organisms, and that also might be leading to intergenerational loss.
So on the other side of that, then people who are eating real food should see a bloom at some point in bifidobacteria, and that wasn't that not the case in my situation.
Maybe, but also it might not. We think a lot of the bacteria that are missing might be present as very, very low abundance seeds in our gut.
Yep.
And we've demonstrated that in animal studies and in human studies, that when we put especially fermented food and a high fiber food diet into people who are not used to it, they can see blooms of certain organisms. And over time, it takes a little while for the gut to adjust, but over time, it can improve their gastrointestinal microbial diversity and lead to the abundance of some of these bugs. But if it's not there at all, then no amount of feeding is going to introduce it. So a lot of clinical studies right now are introducing bifidobacteria, especially this Longum infantis species, into infants early in life. So especially in neonatal intensive care units and in early preterm clinics where we have babies that need that bug, we're adding it into their exposure, feeding them breast milk from their mother, and then that helps to reintroduce it. So it's quite possible, if we're able to do that at a scale that's effective, that in a couple of generations, we might see it being reintroduced, and then beneficial feeding will help to propagate it through life.
Definitely. What's the prebiotic for bifidobacteria? What do they like to feed on?
Bifidobacterium longum infantis basically loves breast milk sugars, the
Mm.
complex oligosaccharides that mother produces in her breast milk that changes over the life of a baby, right? So breast milk is not one composition, it changes constantly, and it varies month to month, and it varies throughout life. So it's complicated. But there are lots of different species of bifidobacteria. And so a lot of the time they like indoles, colorful plants and colorful vegetables and fruits. They can be very full of these kinds of compounds, and they like fiber. So, complex fibers. But, to get that bifidobacterium longum infantis, we need breast milk.
Yeah.
I wouldn't suggest you start trying to consume breast milk as an adult, though, because it won't help unless you've got that bug in your gut.
Yeah. It's sold as supplements, or at least the B longum is sold in supplements, right? But it's getting the
It is.
prebiotic right. And if
Yeah.
it's based on polysaccharides that are exclusive to milk, then we're all out of luck, right? So
Precisely. You need it as an infant, but then it can stay throughout your intestine in life and become part of the beneficial microbiota fed by other organisms. It's not like a panda and it's bamboo, right? It has a broad feeding strategy, but to get it to take hold, get it to be present there, you're going to need early life exposure to that breast milk.
Gotcha. All right, so let's nail down on the built environment. So things like lighting, right? So can artificial lights for, so if you had no lights, right? So sun up to sundown, no lights in the house, but then the artificial lights, whether it's fluorescent, now you're playing with the different spectrum of maybe you've got too much UV, not enough red and near infrared. So is it incandescent lights? What are your thoughts on lighting as a part of the optimized built microbiome?
Yeah, there's been only a few studies that have explored this phenomenon. We know that exposure to direct sunlight through a window, right, or light through a window pane, differs. So, if you're exposed to direct UV without any window there, it can change the microbial community on that surface versus like sunlight that has to go through a window pane versus adjacent areas that are shaded. It can have a different microbial community, and we think that's due to the ability of organisms to survive the UV exposure. But realistically, there appears to be very little difference between spaces that have direct sunlight versus incandescent light on the microbiome. It's far more predicated on what animals, plants, which humans are present in that environment and what surface materials are there. So temperature, humidity, surface material, and occupant are the primary drivers of what microbiome is there on the surface. Light is less important. However, you tell that to anybody that's worked in a basement versus worked upstairs with windows. There's a neurological impulse to having the presence of natural light.
So you wouldn't be too worried about, LEDs versus incandescent after sunset or before sunrise.
Not on the microbiome. It's not going to have a big impact. I mean, it's just the vast majority of bacteria you'll find in the built environment are essentially dead, right? They don't survive very well. You have a nice, warm, moist, nutrient-rich, dense body, and when they leave your body, they suddenly find themselves in a cold, dry, nutrient-poor environment. It's like a horrible desert. Vast majority of them die. The ones that do survive are adapted to that survival environment, and the amount of light isn't going to make a big difference.
Nice. All right. So I think then the factor you left out is wetness, right? So and that's obviously gonna be a factor for microbial growth, probably mold or
It's the
so.
key. It's the key factor. Moisture is absolutely essential. Yeah.
So keeping everything as dry as possible, that's step number one in optimizing.
Well, this is where it becomes complicated. So, yeah, if you have a wall and you make it wet, then the biggest likelihood, if there's nothing antifungal in there, you're going to get molds. Molds are going to release spores. Spores are going to make people sick. We've known about that for 3,000 years, right? We didn't know they were fungi, but we knew that that mold was bad and it made people sick, right? It's even present in the Bible, the Jewish, in the Torah and the first part of the Bible, whatever that's called, the Old Testament. They actually talk about washing walls with blood and water to get rid of the molds which can make people sick. Don't talk about mold, but they talk about the colors. So, we've known about that for a long time. And yes, somehow we still have not enabled ourselves to create plumbing systems or whatever that are totally foolproof and stop that from happening. And that's especially true in very, very humid environments. However, we are working on developing more structures which have antifungals in there. We don't want to get rid of all the fungi. Some of the fungi can be beneficial. We just don't know which ones yet. So we're working on creating these kind of living surfaces which would allow beneficial fungi to flourish and beneficial bacteria to flourish, but not dangerous ones. So it's a real fine balance. We don't have the equation sorted out yet, but moisture is important for beneficial things as well as creating a problem for detrimental things. And we want to balance that equation.
Yeah, definitely, always, right? It's a yin and yang between our existence and the microbes, right? So, like as you mentioned, we live in their world. So how in green spaces is just the greenery resisting? Because if it rains in the forest, there's going to be mold, right? So the forest has to have evolved mechanisms to limit the mold exposure, otherwise it would take over everything.
But I think this is the complexity, right? If I take a mold and I put it into an environment where it has no competition, right? Certain species are going to grow really, really well. Just so happens that the species which can survive and thrive in an absence of competition in our buildings are ones that are detrimental to our health, right? Now, whether that's an evolutionary driver or not, I don't know, but that's what happens. If you take that same bug and you put it into a rich, diverse ecosystem, it'd be like taking a house plant and sticking it in the Amazon rainforest. The likelihood that it's going to suddenly proliferate and take over the Amazon rainforest is very small. There's much more checks and balances, right, in that system that stop dangerous things from dominating or anything particular from dominating. So diversity is extremely important. Maybe I'm not allowed to say that in our current climate, but diversity is incredibly important in our ecological world, right? With diversity, we get a resilience, we get strength of an ecosystem. It stops one thing from becoming dominant and killing everything else. So it's incredibly important that you embrace that. We've removed it for various reasons from our built environment, and adding it back in is very difficult, right? But a child, for example, that grows up physically interacting with a dog in their home will have a 13% reduction in the likelihood of developing asthma, which is very significant, right? So just introducing one source of diverse, rich microorganisms can be beneficial. So figuring out ways to manage that exposure could be vital.
All right, so along those lines, are there green things that you include in the built environment in your own that others may benefit from? To
Houseplants are great, right? There's nothing wrong with them. There's very few examples that we found where there's houseplants are an epidemiological source of pathogens, and a lot of examples where we've shown that having houseplants in the home can actually be beneficial for health. So the benefit outweighs the risk, right? Same with dogs, right? And any kind of animal. So yeah, bringing that natural environment to the home. If you're anything like us, my wife and I are terrible at keeping houseplants alive, but so maybe we're just bringing in new ones each time. But also, if you can, getting outside and having the opportunity to do some gardening, even if it's in a window box, right, with some plants that you grow on your windowsill, is a great thing to do. Having that physical interaction with that plant, as well as being able to breathe in the things it's releasing, are beneficial.
So 100% agree. And actually, that's one reason I'm moving out of Boston and moving more to the suburbia so that I've got that backyard with grass that I can start gardening and do all these
Yeah.
things that I miss out. Not that it's impacting my health here, but it's underrated. I mean, we evolved in nature. That should be some part of our life. So not just, are there specific plants or it's any greenery is going to be
Yeah,
beneficial? Are
we don't
some
have a particular, that kind of tell me exactly what I should buy is just impossible, right?
Yeah.
Because the people that live in Boston are going to have different exposure needs than the people that grew up living in San Diego. And that's going to be very different for immigrants like me who grew up living in a different continent and then came over here and are now exposed to a very different ecosystem. So we actually think your historical, if you will, your ancestral immune exposures can actually be important. There's the Chinese, when they were going from, say, the capital city of Hangzhou out to the peripheries of the empire, would take little tubs of soil with them from their hometown, the place where they grew up. And whenever they felt sick, whether it was just homesickness or gastrointestinal illness, they would take some of that, make it into a tea and drink it. And they believed they were consuming the essence of their hometown. It's quite likely they were consuming things which stimulated the immune system in the way that they had when they were growing up in their hometown, right? So it's a beneficial, learnt environmental immune exposure that they were bringing in. I'm not saying everybody needs to go and grab soil from their hometown and carry it with them, although that may be beneficial. But these are indications that for years we've understood the importance of where we come from in terms of how it trains our immune system and how it keeps our cells healthy. So there could be something in that, right? What you put in your home could be important. We don't have the answer to that at the moment, whether it's hostas or dandelions. I don't know. So the knee-jerk reaction is just to say, just increase the diversity, as many plants as you can, different species of plants. Make sure you keep them alive, as like your pets, and look after that environment. Make it more diverse. Diversity is strength.
Nice. So Are there specific plants that can reduce environmental pollutants? I'm going to look into it, but wouldn't that be a good place to
Um,
start?
in soil and in the air, yes, but it depends upon what environmental pollutants, right? And it depends upon the environment in which they find themselves. In the home, there are people that will tell you that some plants are more beneficial, but it's like they'd only be beneficial if your house was exposed to extremely, extremely high levels of that pollutant.
Mhm.
And then only if you had like an entire room taken over by these plants would you actually see any movement. It's not like there are plants out there which are sucking up huge amounts of air and recirculating it and making it healthy. And but there are people out there that will tell you that that's true, but they're probably trying to sell you the plants.
Yeah. So then the other side of that too is when considering that we're breathing oxygen and expiring carbon dioxide, and plants are doing the opposite. That no matter how many plants you have in the home, you're not going to reduce carbon dioxide levels by them consuming it and then producing
Your
oxygen.
house and your house is not exactly impermeable, right? If you had a completely sealed system like the International Space Station, without those carbon dioxide scrubbers, you quickly run out of oxygen. But you could theoretically, with enough plants, create a system that was entirely self circulating. But you need a lot of plants. Yeah.
Yeah, isn't that with the, have you heard of Terra Preta? I think it's the Amazonian soil that I guess it regenerates on its own, and you don't have to add fertilizer. And
Well,
it's
soil is another entire question, right? Yeah,
yeah,
soil is a rich microbial ecosystem, but without a plant inputting carbon into it, it can quickly become self-cannibalizing. So you do need an entire ecosystem. For soil, you need the plant, you need the fungi, you need the bacteria and the viruses all interacting to create a self-sustaining ecosystem. A soil by itself that was self-sustaining would have cyanobacteria, bacteria that can take light energy like a plant and make it into sugars, but they'd only be on the surface. And then you basically created a very, very specific ecosystem that's surviving in a stratified form. I can get into that, but that becomes complicated.
so then even soil, you can make the argument that there's going to be a spectrum of what's optimal soil for greenery to grow versus, nutrient dilapidated and just right. So
Yeah, 50% of our soils, our agricultural soils, are already nutrient and microbially dilapidated. And by 2050, we expect that to get up to 90% of our soils. So we are in a critical inflection point in soil care. We've actually opened a new soil health center at UCSD, which I'm co-running, that's predicated on trying to do exactly what we're doing in humans, get the beneficial microorganisms back into their experience, but we're doing it in soil, getting the beneficial microorganisms back into the soil to help to restructure the carbon cycle and nitrogen cycle of soil to make it more self-sustaining and make it more resilient to climate change and industrial agriculture. Because without that, we're going to run out of soil, and soil is feeding 100% of our population.
yeah.
So if you have damaged soil, you're going to get damaged food security. If you have damaged food security, then you get mass migrations, riots, and the destruction of humanity. So it's kind of important.
Yeah. So is a part of that story composting then? I mean, should we all be composting and
I
throwing our
mean,
scraps?
composting's great, right? But we find that it's very difficult for a lot of the, especially poorer people in our community, to be able to, in the world, to be able to compost. So, for example, when we go into sub-Saharan Africa, where 900 million people are actually fed by essentially smallholder farms, mostly female-run smallholder farms, um, the the fiber, if you will, the organic matter which they produce during the production, they need to act as a fuel for keeping their houses warm, to feed the animals. Very little of that organic matter can go back into the soil or can be composted, right? Because they use it in everything. So in those situations where food insecurity would lead to absolute global chaos, this is essentially why we need US aid. I won't get political at the moment. In those environments, a lack of different strategies to improve food security is going to lead to a massive problem. So we're going in and we're creating synthetic probiotic communities. So communities of bacteria from that environment that we construct, we grow, and then we redeploy into those soils that can actually be effective in helping to restructure the soil ecosystem and make it healthier and more resilient, so that we don't necessarily always need composting. Obviously, ideal situation is we take composting and we add it back into the soil. We use regenerative farming practices, we put it back into the soil. But it's not always possible. So we need lots of different strategies to make that effective.
So the probiotic that you're talking about, what would be the prebiotic then in that case if it's not composting and what?
The prebiotic is what the plant pumps out. So just like mother's breast milk, a lot of the soil bacteria become dependent upon the carbon exudates. We call them exudates. It's just what the plant excretes from its roots that the plant pumps into the soil. So that's why you need the plant and you need the microbiome in the soil. And the plant's roots pump this out, and all the bacteria congregate around the roots, they munch it up. And then when they're munching that up, they consume other things around the soil and make that bioavailable back to the plant, right? So the plant is essentially seeding the soil with beneficial nutrients to make the microbiome thrive, and then the bacteria pump the beneficial products back into the plant. Exactly the same as what happens in our intestine with our microbiome, right? So we're trying to understand that cycle and make it more beneficial. The unfortunate side effect is if you pump the soil full of nitrogenous fertilizer, the synthetic nitrogenous fertilizer, it feeds the plant, but it kills the microbiome. And it makes the microbiome very, very dependent upon that source of nitrogen. And the microbiome now is no longer interacting with the plant. And so it starts to become very non-diverse, entirely self-reliant upon that nitrogen, and it starts to die, right? So you have to have a constant replenishment if you're going to use that. The idea, what we're trying to aim for is where we only have these microbial probiotics and we don't rely on nitrogenous fertilizers, we don't rely on phosphates, we try and avoid the use of herbicides and pesticides. But it's not, we can't change an entire global economy overnight, right? So we're working on trying to develop systems which will integrate or can be used alongside those damaging practices in order to mitigate their impact.
Is the soil microbiome similar to the human microbiome? Like, for example, high fiber diet, you're going to get short chain fatty acid producing bacteria. You're going to minimize the growth of the enterobacteria, protein digestion. So on a high nitrogen fertilizer containing diet, I'm thinking a lot of enterobacteria, a lot of proteobacteria, and low fiber degraders, right? So
ostensibly, yes. I mean, you do see short chain fatty acid production in the soil, but that's not quite what's important, right? We see other kinds of compounds which are relevant for promoting plant growth and for being able to degrade certain complex phosphates, which are only present in soil. I remember the soil microbiome is the most diverse microbial ecosystem on the planet. There is nothing more diverse in it. The human gut, it pales in comparison, right? So we're talking about an incredibly complex ecosystem. And so it's like the human gut on massive steroids, right? But yes, exactly the same principle. If you just eat saturated fatty foods that are processed, you're going to get lots of proteobacteria, a total loss of all the Bacteroides, some of the beneficial bifidobacteria, the lactobacillus are going to go away. You're going to get a very undiverse ecosystem. Exactly the same is true in the soil.
Interesting. All right. So going back to the built environment. So carbon dioxide, if carbon dioxide levels in the house are, I know you were saying if you're on the space station, but so I'm moving to Austin and, I've got to make sure that my house is pretty well sealed so that I can cool it, right? So then monitoring air quality and carbon dioxide levels is going to be important because it's a part of the overall sealed structure, right? So, is carbon dioxide, a part of the built environment, keeping that at a certain level? If so, what level to minimize microbial growth?
In your apartment in Austin, even if you've tried to seal the building, the air is still coming in from outside. It's just being processed. So you're never in a situation where you're recycling the air inside the building in a way that would ever lead to a significant increase in carbon dioxide concentrations. So we even when we look at positive pressure rooms inside hospitals or negative pressure rooms inside hospitals, even when we pack it with people, we see only a small increase in carbon dioxide concentration in the atmosphere. But we actually demonstrated you can use the carbon dioxide concentration in the atmosphere to predict how many people are present in the room, because it does go up a little bit, but never to dangerous levels. It just can't. I mean, there's just too much air exchange with the outside. So it's something you can look into, but it's never going to be a major threat. What you really do need to focus on is the fact that you're dealing with an environment which is dynamic and has a lot of opportunity for change, right? So putting in beneficial things will not necessarily worry about oxygen and carbon dioxide, but will improve what your body is exposed to in terms of the antigens, in terms of the reduction in some harmful gases, that kind of thing. That's what we're most interested in.
So air purification then should be a big part of the story.
Yeah, but you want air purification alongside. If I just put an air filter in a room, I'll get rid of the bacteria and the viruses and the fungi. If I take an infant and I put them in that room, they're never going to get any kind of microbial exposure, which would actually be beneficial to their health.
Hmm.
But if I take them and I put them in a room with an air filter, it gets rid of the pathogens they might be exposed to. But then if I just stick a dog and let the dog lick the baby's face, and I put loads of plants around the room, I'm providing further stimulation that child can then get in order to improve and develop its immune system. Same is true for you, right? You cannot just purify the air and hope for the best. You need a rich, diverse exposure to help you.
Definitely right. So just wait, pushing back on that a little bit, then couldn't I just go outside and then
Yes,
outside I've got the
going
green
outside is great.
yeah, and I've got the antigenic stimulation, whereas then I can go inside and then basically let my immune system relax a little bit because it's not constantly bombarded.
Absolutely. It's just unfortunate that most Americans spend 90% of their time indoors.
Yeah, definitely.
Going outside, and if you look outside the vast majority of American, even suburban environments, there aren't a lot of places to walk, right? It's a car-driven society. Most places don't even have, sidewalks. We in the urban environment, it's even worse. So this is a real problem. We need to provide people with access to those spaces, and the only way to do that is to either build them or bring them into the home, like we did with bringing soil into a kid's experience, or taking the kids to the forest, or making the building itself more biologically viable. That's the way that we've overcome the limitations of our current societal norms.
Definitely. All right. And then, so in terms of the wetness, so then that gets the humidity, right? So is there a humidity level that once the built environment gets above a certain percentage that, like how wet, like how humid? Below
This is
45%?
yeah. There's an entire there's an entire society, indoor air society, that's focused on this question. The majority of it is actually keeping the humidity at a level which makes us feel comfortable,
Hmm.
right? And as long as we feel comfortable, it's very unlikely the humidity is going to be high enough to allow moisture to form on walls, which will cause fungi to grow. But again, there are ways to mitigate that fungal growth, either with the introduction of antifungals into the building material, or potentially creating a living structure in that building, which prevents the growth of fungi, or prevents the dominance of particular fungi, even when it is wet. But those are ongoing research topics and not necessarily developed products.
Gotcha. All right. So the Amish story is so they're eating whole food. They're not, you
So are
know,
the Hutterites.
hmm,
Both of them are eating very healthily. Farm-based products, lots of fruits and vegetables, traditional, lots of potatoes and animal products. So traditional, southern northern German diet. But yeah, one has the children growing up and being exposed to that farming environment. The other one doesn't.
gotcha there. All right. So another one is since I think you're the director of planetary health, on your long list of accolades,
I
uh,
wish sounds amazing.
or
Planetary health. I don't know.
something along those lines. I have it here in my notes, but wait,
I can be the planetary health director. I'll take on any
maybe
role.
not planetary health. I'm overstating it. Which affiliation I'm talking about.
We
I'm sure.
ran an Earth microbiome project.
There
I
you
can help.
go. Yeah, yeah, yeah. Sorry, I gave you the glorious, right? So,
I don't know. Planetary health sounds way better. I'm taking it.
all
That's
right,
my
cool. So when you consider that our microbiome co-evolved over, 500 million years of human existence, right? So, or hominid existence. So are we stuck on this planet never to travel and step without, breathing apparatus? Because if we do, our microbiome isn't adapted, and then any antigen may kill us unless we've evolved the technology to adapt
no,
to
no.
that too.
I don't believe that. I mean, our bodies are highly adaptable. Hominids are only about 5 million years old, but yeah, 500 million years is everything you can see with your eyes. So
Mm.
from fish and reptiles to amphibians and plants, that's all about 500 million years old. But yeah, so if you take a human and we put them on Mars in a controlled environment, there's a possibility there might be an antigen there which might make us sick, just the same as when I moved to America, I had allergic reactions to different types of oak pollen. It's now gone away through dietary interventions, etc. But it's a problem, right? When you move to a new environment, you're going to get new exposures. But there's no reason to believe that living on a different planet would lead to any significant shift in our immune response that would cause detrimental health impacts.
So just to play the devil's advocate, so when the Europeans came over and the Native Americans weren't familiar with those microbes, wasn't there a wiping, wiped out
That's
big
that's a
broad?
pathogen. That's epidemiology, right? And so it's possible if we landed on another planet that had rich life, that there'd be a pathogen there that would kill us off pretty quickly. I mean, that's H.G. Wells and War of the Worlds. Those aliens couldn't survive it. The Native Americans similarly impacted. When the Dutch came back from China and brought back infections, we caused plague in the 13th century. Same thing happened in the 2nd century Roman Empire. Those are epidemiological disease problems. That absolutely could be true.
So then, if there are aliens that exist, one reason that they're not stepping foot or we don't see them is because they're not worried about getting microbial diseases because they haven't evolutionarily adapted.
So there is a interesting sideline. We think that xenophobia may have evolved as a mechanism to prevent us being exposed to pathogens from other people. So if you're in your isolated tribe in the jungle, right, you actually become quite immune over 1 or 2 generations to anything that's circulating in that population. Then a foreigner comes in from a tribe over the other side of the mountain and wants to trade. Well, they have other pathogens. They bring them to you. Your tribe gets sick. So that would rapidly select due to lack of progeny. That would rapidly select for people who were wary of other people who weren't part of their tribe, right? So xenophobia may have evolved because of microbial exposure, and it's possibly true for alien societies. Maybe the reason we don't see alien societies is not because of the almost impossible to imagine technology that would require us to move at light speed or close to light speed, and the extraordinary differences even within our own galaxy that would preclude us from seeing anybody else, but because they don't want to get infected.
Well,
H.G. Wells star.
yeah, I mean that's
But
a whole nother conversation because you're making the argument that they're coming from somewhere else when the Earth is what 4.5 billion years. Who's to say they're not here and that our small visible spectrum that we can actually perceive, maybe they exist in portions of it that we can't directly detect with.
that's absolutely possible, yeah. And it only takes around 50 million years for the surface of a continent to be so significantly shifted that it's hard to detect anything else. And our fossil record is so sparse that if a population of highly technologically evolved people suddenly appeared and then was wiped out in, say, 50,000 years, which is likely what's going to happen to us, it's quite probable you would not see any signature of that in the fossil record. Or the only way you might see it is if you were looking for certain chemical changes that could exist in isotope ratios. But you'd actually have to look at it in very particular locations, and that might be the only thing that you'd see, and it'd be very hard to interpret it as the rise of a civilization. So yeah, absolutely. Stuff could have happened 50 to 100 million years ago that we would have no understanding of. The aliens might have arrived, just before the dawn of the dinosaurs, or even during the dinosaurs, set up an entire culture and civilization, and we'd never know about it.
Yeah, definitely. All right, so how much is fiber, soluble fiber, because there is a gut-lung axis, how much of that is a part of and I get the asthma story and the dust transfer, right?
Yeah.
But did you adjust for fiber? Because, so there's at least one published study that I know of where soluble fiber supplementation improved survival in mice infected with influenza.
Yeah.
So with
That's immune interaction. So if you have high levels of fiber in your diet, you'll have an increased level of short chain fatty acid production, an increased level of bile acid deconjugation, which will lead to alterations in your immune response and make you more likely to be less susceptible to extreme inflammatory reactions in the presence of a pathogen.
So is that part of the story in the Amish Uh,
Quite possibly the gut microbiome axis and the gut immune axis is absolutely essential. But as I say, in that population, both of them are consuming very similar diets. So it's less likely to be a particular driver there. It's more likely early life environmental exposure is shaping that paradigm. But we do know that, for example, we've demonstrated that certain lactobacillus species that we can isolate from the lung can be highly effective in helping to treat things like chronic obstructive pulmonary disorder or lung fibrotic diseases. And that appears to come from the gut into the lung, potentially via esophageal transfer into the lung environment. And we believe that might be driven by particular diets. But again, a lot of conjecture.
Interesting. All right, wait. So there's also a few published studies on soluble fiber helping to, and I'm going to say detoxify, but helping to protect against microparticle pollution, which implies that it can improve lung microparticle. I don't know if you're familiar with that, but there's just a few
I'm not
studies.
familiar with that. We've looked into the microplastic exposure, and we don't see any significant association with dietary health at all. In fact, when we look at microplastic exposure in the ocean or in the environment, we can't seem to see any particular impacts upon animal or ecosystem health. But depends what microparticles. If those microparticles were like cadmium oxide, then that might be a more bigger problem. But yeah, I mean, again, if you have a healthy immune system and you have a healthy body generally with a lower antioxidant level, which is generally associated with improved immune responses, which can be modified and managed by a beneficial diet and a beneficial microbiome, then your body's going to be more resilient to a lot of things.
Definitely. Isn't there some evidence that the amount of particulate it's less than
PM
2.5
2.5s.
yeah, isn't it going to increase over time because of, global warming and all of the other stuff, fires and
Ostensibly, if there are more fires, yeah, but PM 2.5s and PM10s are heavily associated with pollution in any context. So when you burn fossil fuels, when we burn wood, any kind of air pollution, we can get particulates that build up in the air, and they are definitely damaging to health. We've demonstrated that, and there was one legal case in the United Kingdom where they were able to sue for a child's death by demonstrating that child would not have died had they not been exposed to that pollution.
wow.
Right. So, and that's going to happen more and more and more, where it's going to become a legal necessity to control that pollution in order to prevent lawsuits.
So in that context, no amount of a healthy diet, if PM, particular pollution is increasing over time, there may be a threshold
Yeah,
where you just,
but bear in mind, it's not going to increase like it's not going to become a global phenomenon, right? It has to have a point source. All particles have to have a point source. A forest fire is a point source, right? It can be a very generalized point source, and if you're near it, it's going to make you very unwell. Yeah, at some point, I cannot make you impervious to damaging environmental stimuli, right? If I put you in an environment with a car is pumping out pollution into your face, no amount of microbial support is gonna stop you from getting very, very sick and probably dying.
yeah. All right. So future or current projects that are unpublished that you want to talk about and future projects that are on the,
Oh man, I have 47 ongoing research themes in our lab, and I can't choose my personal favorite baby. But I think one thing we're really, really focused on now is trying to improve women's health through modifying the gut microbiome and the vaginal and cervical microbiome to essentially reduce women's female-associated disease causes, and also improve menopausal symptoms and improve aging through that framework. So that to me has been something incredibly interesting and one thing we're focused on.
but
We're also very interested in the gut-brain axis. I work a lot in depression and anxiety. We're starting clinical trials focused on trying to reduce depression and anxiety by altering the gut microbiome with particular probiotics that we've isolated from healthy people's guts.
healthy non-depressed people. So taking
Mhm.
probiotics from them and then, but then you
Putting
need
them in.
the prebiotic in that case to feed those probiotics. Otherwise, it would just go away, right?
Yeah, so actually we actually don't think they need to be alive. So we've run our preclinical studies in rats. These bacteria, even when they're dead, are as effective at treating depression as Prozac and ketamine.
How's that possible? They're going to be in the gut, dead, not making any
Things don't
metabolic.
need to be alive to interact with the immune system.
Hmm.
So I can take a dead bug, and the proteins and sugars on the outside of its cell, or even on the inside of its cell, will be seen by the immune system. The immune system doesn't recognize an entire bacterium. It recognizes chemical signatures on the surface of its cell, or things that it pumps out, like hydrogen sulfide or lipopolysaccharides. These are things which bacteria pump out. Like, so flagellin, lipopolysaccharide, and hydrogen sulfide are predominantly produced by things like proteobacteria, and the immune system sees them as pathogen-associated markers, and they cause inflammation. I don't know why I've got confetti. Apologies.
Me neither.
But that's an important piece, right? It's things don't need to be alive. Chemicals can stimulate the immune system. So if you have the right kind of bacteria with the right kind of compound on its surface, alive or dead, it will produce an immune response, which can shape how your brain functions.
So the depressed people are missing the dead or alive
They're
bacteria.
missing that bacteria. Whether it's alive or dead doesn't appear to matter. They're missing that bacteria in their gut.
And are they fiber degrading bacteria? Because that would be my bet.
Some of them will degrade certain. Well, this one, the one that we're focusing on, does degrade certain fibers, but it's more related to polyphenols. So if you have polyphenols in your diet, it will stimulate the growth of that one. But again, it's like the bifidobacteria story. If it's missing, no amount of polyphenols are going to bring it from a non-existent to an existent state, right? It needs to be introduced.
So I was going to ask about fecal pH, right? So, but the probiotics that you're mentioning probably have nothing to do. It doesn't matter. They're just dead. The immune system is triggered. But I think, what are your thoughts on fecal pH as an underrated aspect of optimizing the gut microbiome? I mean, you can eat a high fiber diet, but not every study shows that even with a bloom in short chain fatty acids in fecal samples, that fecal pH goes down by a significant amount to potentially optimize gut microbiome composition.
Yeah,
So
it goes. It only fluctuates by less than 1 pH point, right? We're talking about a change from, 5.8 to 6.2 or 6.2 to 6.4. It's that kind of range of fluctuation. But yeah, fecal pH is very indicative of the antioxidant environment or the oxidative environment in which that microbiome lives. And the oxidative environment in which a microbe lives is entirely responsible, or very responsible, should I say? I mean, not entirely responsible for its metabolic activity. So what it can eat and what it poops out will be very dependent upon the pH. But the fecal pH is a proxy measure of what's going on in a part of the intestine. We, realistically, to understand it fully, we need to get pH measurements from the duodenum and the stomach and the esophagus and the small bowel and the distal sigmoidal colon and you need everywhere because the ecosystem changes as it goes through the gut. But yeah, it's a good proxy measure that people don't always look at. But the same is true for gastrointestinal transit. We now measure transit sometimes, but not always. And as we get older, the reason we tend to have more gas and we start to fart more is because our gastrointestinal transit slows down. And so our body is actually processing the food more consistently, and that produces more microbial activity, which produces more gas. It's an interesting side effect.
in the wrong direction, though, you get probably more protein digesting,
You do get more protein digestion, but it depends upon your diet, right? If you have a high fiber diet and you have a consistently high fiber diet in a 20-year-old and a consistently the same high fiber diet in a 50-year-old, you're still going to get a slower transit time and a significant increase in gas production. Now, it's possible that you could, for a high fiber diet in a 50-year-old would lead to a significantly faster transit time than a low fiber diet in a 50-year-old. But there's some part of aging that we don't fundamentally understand in terms of gastrointestinal transit.
So fecal pH can actually vary a little bit more. In centenarians, I think there's one study where it was close to 8. But granted, this is, the fecal sample. You don't know, is that a measure of the whole, the
Yeah,
colon or not?
and I always, yeah, I'd love to take the centenarian studies are great, but you're dealing with a group of people who are living a very different life and have lived it for a long period of time. And so, we don't know which part of their life may have resulted in the features which they have as a 105-year-old. It could have been what they were exposed to when they're babies. or what happened when they were teenagers. It's until we can fundamentally replicate that in another individual and demonstrate over a very long period of time that it's associated with longevity and everything else, it's going to be very hard to prove it. We can do some of that in mice, right? But in humans, we need, this is a call for action, right? If we're ever allowed to do science again in this country, or maybe all the scientists will have to leave and go to China and make China the most dominant superpower on the planet. If it's a threat. If we can, we need longitudinal studies where we study populations over decades, so we actually understand at which point we can have a real significant impact upon longevity and upon healthy aging. That's incredibly important. Right now, we're just making suppositions and hypotheses.
100% agree. I wasn't suggesting that centenarians having a pH of 8 may be beneficial for their longevity. In fact, I was potentially arguing the opposite. That's a consequence of a longer life having a higher fecal pH. Along those lines,
Maybe, yeah, that's a really good point. I didn't think about that, and it's possible that there's some physiological feedback mechanism which would lead to that feature. Absolutely.
so along those lines, there's a few fecal pH studies, at least in Parkinson's patients, where fecal pH is relatively high, as close to around 8. And the reason I raise that issue is because uh, so you mentioned fecal pH being a part of metabolic activity, right? And anoxia versus anaerobic, or anaerobic versus aerobic conditions. But fecal pH can also impact bacterial enzymatic activity. So now
Well,
you're getting,
yeah, I mean that's all part of the same piece, right? So pH is a function of microbial metabolism, which is a function of the oxidative environment. You're right, the amount of aerobic and anaerobic activity, but also the number of anions and protons in a system, and the number of ionic exchanges, which is all a function of the metabolic environment, which is a function of that system. I mean, it's a great example. If you eat more fiber, your your antioxidant level inside your body will be significantly increased. And I love that this is a great example. Um, when you look at antioxidant activity, our bodies have an innate antioxidant capacity, right, to eradicate free radicals and oxidative stressors, which will damage our neurons and damage our cells and stop our functions. If you eat a crappy diet, right, high saturated fat, high sugar diet, you do very little exercise, you live in an apartment building in a polluted environment, you are constantly putting your antioxidant capacity to the absolute test, and quite often going far beyond its ability to control free radicals and control oxidative stress. Um, if we can take those people and just replace one of those pieces, like a healthy diet, or improve their air quality that they're breathing, we might be able to bring it below their oxidative stress threshold and help them to live a healthier life. I just think, our modern lives have so many threats that it's constantly pushing us above that threshold and preventing or damaging our ability to control the damage that oxidative stress can do to our neurons, our body cells, our liver cells, our kidneys. And if we could control that, we'd have a much better chance at living a healthier, longer life.
definitely 100% agree. But where I was going with that is bacterial metabolites that could impact the brain. For example, quinolinate. I'm sure you're familiar with the work on quinolinate being neurotoxic, even in Parkinson's, right? And it increases during aging. So most people are focused on quinolinate coming from the de novo NAD synthesis pathway, so degradation of tryptophan all the way
Sure.
down to the quinolinate. But quinolinate can be produced by gut bacteria by aspartate oxidase, which is sensitive to a high fecal pH, which argues that low fiber diet, high sugar, Western diet, high fecal pH, increased bacterial activity and conversion of aspartate into quinolinate, potentially more neurodegeneration. Nobody's
They've
looking.
said the same thing about kynurin, though, right? And
Yeah,
and the animal studies have in sometimes supported those statements, and other times have shown them to be highly variable. And I think we need more studies in humans to demonstrate that there's a cause and effect pathway there. More protein can lead to a significant increase in hydrogen sulfide production through short chain fatty acid production from protein fermentation, right? It's the wrong kind of short chain fatty acid production can cause that inflammatory response. But you're absolutely right. There are many of these processes which might be linked.
gotcha. Well, thanks, Jack. That was great. I appreciate you coming on. Hopefully, you can come on at another point and we chat some more about the microbiome.
Yeah, sounds good.
All right, cool. Bye, Jack.
Cheers.